Hi, Influenza Update. And here's our little influenza rotating virion on the top. So, influenza viruses and Orthomyxoviridae in general are not just viruses of mammals. The four influenza A, B, C, and D are shown here, and they're in this clade that mostly infects chordate animals, and a lot of those are mammals. But if you look a little further, you find all sorts of strange things. So this big red clade up here is actually looks like two clades. This has several strange viruses in it that are Orthomyxoviridae. So they're put together like an influenza virus, negative strands, segmented, multi-part replicase. But these are going to infect things like Xene coelomorpha, which is a very strange little invertebrate clade, arthropods, mollusks, Cnidaria, which is like jellyfish, and ctenophores, which is like comb jellyfish. And so this is probably showing us where the actual root is of this clade. It's very interesting. Then you look down here and you can see several viruses that infect mammals, for example. But if you go zooming into this thing, you see a bunch of Togotoviruses. That's the THOGO. And those are associated with primate infections, and those are something that we're always watching for that could spill over to humans. Those Togotoviruses, most of the group that's around them actually appears to be native to arthropods. And a lot of these are like biting insects, and so that's a way that they may be able to transmit. So yeah, a lot more arthropod viruses. Then look at this other clade. This is the other half of that same thing, but it was too small if we just put it on one page. You can see that most of these have this sort of little blue square right down there. That is an arthropod native clade as far as we can tell. A few do jump over to chordates, and it may well be because of biting insects, mosquitoes, and things like that. So, a lot of orthomyxoviruses out there, and while the genome structure and mode of replication is going to be common to them, they have a lot of surprises in there, and a lot of the structural proteins are not known for some of these others, but you would expect them to be quite different. There's quite a few of these in fish, and tons of them in insects and other arthropods. Okay, next concept, which is new but actually ancient: antigenic shift versus antigenic drift. Since orthomyxoviruses are segmented, their genome is broken up into little RNA segments. If you ever get a situation where two different orthomyxoviruses, same species or not, have infected the same cell, theoretically, their segments can swap around. They can rearrange, and as long as the virus comes out with a complete set, there's at least the potential for it to have, for example, a totally different hemagglutinin to the one that it was normally traveling with. And hemagglutinin controls tropism, and so you can potentially get in one fell swoop a virus that's able to infect a totally different host, but carry all the other genes that work together and make it pathogenic. This is what is thought to have happened in the swine H1N1 pandemic influenza back in 2009. It's also thought to have been the mechanism behind some of these ancient things, like it was called the Spanish flu, but it occurred right around the end of World War I. So that's antigenic shift. That is like a one-for-one swap. It's almost like a recombination, but recombination involves copying one strand, switching to another strand, and incorporating genes that way. This is just swapping segments, basically. Antigenic drift, on the other hand, is the process of gradual mutation that accumulates through time and is selected by natural selection. Both of these are somewhat unpredictable, but the antigenic drift moves much slower. So when you think of shift, think of a big shift, like you push the shift key on your keyboard to make a lowercase letter into a big letter. There you go. Antigenic drift, think of that as just formless, unguided, one nucleotide tide at a time drift through sequence space. They can both have big impacts on whether or not you get sick and whether or not your vaccine is going to work this year. But they're very different mechanisms. We would associate shift with things more like pandemics, like big changes that lead to unexpected results. Drift is kind of what we're always dealing with. That's like the bare minimum in terms of problems from influenza viruses. Okay, let's talk the PA protein. PA has several domains. You can break this down into an N-terminal domain, a C-terminal domain, and a linker. Linker doesn't do anything, it just joins those two together. You can also break it down into a number of interaction sites. So everything in this part of the figure is showing an interaction with something. Like this HCLE, C14 ORF 166, to its friends when it's in trouble, I guess. That is a protein that would normally associate with polymerase 2. And by binding to this, the PA effectively has bound indirectly to polymerase 2, and it can then just grab messenger RNAs as they are being synthesized and steal them and decapitate them and use them as its own primers. Yeah, because that's the kind of virus influenza is. Up at this end, we've got interaction sites. We've got two nuclear localization signals to get it into the nucleus via importin. And we've got interaction sites with the other two replicase components. But there's a weird thing that happens sometimes. So you can translate the gene as normal and you get this 716 amino acid product, or you can translate the gene and you can frame shift and stop early, in which case it'll produce the protein called PAX. Now, PAX, like the PA N-terminal domain, has the complete endonuclease. So it has two magnesium binding sites. It can chop RNA into little pieces, or rather, it can cut in the middle of an RNA, which destroys that RNA. Whereas the full-length PA is able to interact with PB2 and PB1. It's able to pass off the stolen cap from the cellular RNA to the rest of the polymerase, which is then able to extend it as if it was a primer. PAX can't do that because it's missing the interaction domains that would attach it to PB1, for example. So what PAX is thought to do is it just runs around the nucleus, and anywhere anything is being translated, or anything is being transcribed, it can bind to polymerase 2 and destroy messenger RNAs. So it's just going to go around destroying messenger RNAs that are not coming from itself. Now, the virus messenger RNAs are going to be packaged up, and they are going to be able to escape this fate. PAX, a powerful force in host shutdown. It doesn't shut down the host as completely as picornaviruses did, but it limits the ability of the host to produce things like interferon messenger RNAs. The frameshift that makes PAX, the short version of PA, is actually a +1 frameshift, and it's a weird looking thing. It works like this. So instead of a pseudonaut, you have nothing. The thing that is controlling the rate of the ribosome is not it bumping into an RNA secondary structure. It's the rarity of this particular arginine codon. And so the codon is going to bind to a CGU over there. And there's just not much of this particular tRNA with this arginine on it available in the cell. And so because of that, if the ribosome gets, I don't want to say bored, but that's kind of how I imagine it in my mind, personifying it. If the ribosome is not able to get the correct tRNA, then occasionally, if it happens to be on a very little slippery site, so this is UUU, kind of like half the slippery site that we got in some of the other viruses, then it'll actually slide, but it won't slide back, it slides forward. So it's a +1 frame shift. And so the dots are where it was, and the blue thing is where it is, and this is the big ribosomal subunit. So yeah, +1 ribosomal, programmed ribosomal frameshift, programmed by having a very rare codon that's needed right there. Because of this, when it shifts forward 1, now it's not trying to read CGU, which is what it was before. Now it's trying to read GUC. That one's very common. That one puts in a valine instead of the arginine that it was actually looking for. So we've shifted our reading frame, and we're going to run into a stop codon very soon. But that will produce the PAX, which is like the free agent version of PA that just destroys RNAs but does not pass off the cleaved RNA terminus for use as a primer by the rest of the replicase. All right, here's a little more on the replicase, and hopefully this will help it make sense a little better in your mind. So when PA cuts a little bit, so PB1 may be already holding on to the cap, and PA is going to cut about 12 nucleotides from the cap. Once that happens, and actually it has to be about 12 nucleotides because the distance between PA and PB2 on this complex is about 50 angstroms, and it takes between 10 to 13 nucleotides to get across that distance. So yeah, that makes sense. And then the cap, if the cap is still being bound here, we're going to thread that primer down here. The N just means that it doesn't really matter what nucleotide this is. It's any nucleotide. That's what it's saying. PA has a preference for cutting at Gs, but it will cut after 12 nucleotides no matter what the sequence is. It's just if there's a G that's around there, it'll slide a little bit and prefer to cut after the G. And if it can get an A and then a G and then cut, then great. That's the highest affinity cleavage that it will make. This primer is then just long enough, so it's 54 angstroms to go from PB2 all the way down this channel in PB1, and that takes 12 to 15 nucleotides. Yeah, so you see why the primer tends to be 12 nucleotides long? It kind of has to be, right? To stretch from the thing that's holding it to the thing that's cutting it, and then from the thing that's holding it to the place that's using it. That's just how far away those things are. And so that's what the virus has evolved to do. Now, the virus is going to be copying the virion RNA, the vRNA, because we're making a messenger RNA, which can be like a plus strand copy of the minus strand, and this plus strand can then be used to translate various viral proteins. So we're going to extend this just like that, and that's great. The template is going to come up this way and is going to end up going out here. Nucleotides are going to come in through this little channel on the side, and the vRNA actually wraps around. So imagine a big long loop, and it comes back up in here. So it's going to make a little panhandle, and it's going to bind to itself, and this is going to be the thing that is found in each segment. And then just after the panhandle, it folds up and makes a tiny little stem loop, which some people have called a corkscrew. And the tiny stem loop actually binds to a 5' binding pocket right over here. And so this is what's going to activate PB1. And this is how it works. It's like the key in the ignition. And then it doesn't show it, but there's a big long loop here that's the rest of the gene. And this is the other end of it. And we're going to be copying from the 3' end, so this is the 5' end here. And when we finally have pulled all of this stuff through, then it can get to the end and potentially copy that. All right, these are unchanged and very helpful. And let's get to the end. Here we go. So the other, and this is the last thing that we're updating, is NS1. So it's a non-structural protein, non-structural protein 1, but they just call it NS1 in the field. It looks like this. It functions as a dimer. There's actually 2 of those RNA binding domains, and the 2 effector domains sit farther apart. The RNA binding domains interact with each other. The RNA binding domains seem to bind to the viral RNA species. And this seems to work really well at protecting the virus from recognition by some of your favorite proteins, blasts from the past, things like RIG1 and MDA5. So NS1 is going to bind to the end of its own RNA. And so now there's nowhere for the recognition domain of RIG1 to bind. It's also going to interact with protein kinase R and some other kinases. When it does these things, so RIG1 is not going to phosphorylate NF-kappa B or IRF3. They're not going to go to the nucleus. NS1 is going to control eukaryotic initiation factor 2A, and it's going to bind to eukaryotic initiation factor 4G. These are going to enable the virus to cheat a little bit, so they make it harder for cellular genes to be translated. And NS1 will also bind down here if it gets into the nucleus, and it will inhibit the poly-A binding protein number 2. So that's going to be needed to export cellular messenger RNA. And so we're going to trap any newly produced RNAs in the nucleus. So the virus NS1 protein is shutting down the signal that would lead to interferon production. It's shutting down some of the messages that could also lead to mRNA interferon production, type 1 interferon production. And it is going to inhibit the normal mechanism which would lead to messenger RNAs coming out of the nucleus, even if they are made. And those together with PA greedily stealing messenger RNAs for things like interferon, type 1 interferon, that's one of its favorite RNAs to steal. That just means that the virus has a good way to block the cell from recognizing it, and so it doesn't have to deal with all that stuff. It's very dependent on one gene, the NS1. There are a couple other things that work a little bit. NS1 is also going to, here we go, interact with several things. So here's the part that interacts with EIF4G. That's the part that was cleaved in picornavirus infection. And CPSF4, you remember CPSF6 from HIV. This is going to be a factor that's going to go into the nucleus and be involved in polyadenylation process. And several other viral proteins or cellular proteins. It'll also interact with protein kinase R here. This is an RNA-triggered protein kinase that is very useful at setting off antiviral responses. NS1 kind of does it all. Yeah, it's not perfect at any of these things, but every little bit of this helps the virus to do well. Okay. And that's the end of our update. I hope that was useful.